Those are the pedicels. So you know this is filtration side, this is plasma side. Because of the proteins that are present in the plasma, it also has a more floccular look. You can see more solid material present in the plasma rather than present in the filtrate. All right, so now let's look at some of the forces that move fluid across from plasma into the filtrate. So may I race to the right of the capsule here? You ready? I'll keep this picture up. I'm going to erase that diagram. Okay? Anybody else?
I thought you said so it was the 3 major factors that get out of the filtrate into the blood?
Does the
Out of the
polycyte secrete
filtrate into
anything?
the blood.
No, they are capable of contracting, and when they do so, they make the spaces more narrow, so it decreases the amount of filtrate that forms, but we'll cover that. They don't really, the filtration is just determining what particles in the blood plasma make it into the capsule. So it's separation on size and those charges. All right, so where does that force for filtration come from? Hydrostatic pressure. It's opposed by colloidal pressure, all right, the presence of the proteins. The osmotic pressure, we talked about this with the lymphatic system. Remember, we had the hydrostatic pressure pushing fluid across the arterial end of the capillaries, but our large proteins like albumin aren't large enough to cross over. And as we lose fluid, the osmotic pressure gets higher and pulls fluid back, prevents more fluid from leaving, and starts pulling fluid back into the venous end of the capillary. So we see some of that opposing force here. So drawing again a smaller version of our capsule. So our primary force is hydrostatic pressure pushing fluid out of the glomerular capillaries into the filtrate. And so that's identified as capillary blood pressure or capillary hydrostatic pressure. And it's around 50 to 55 millimeters of mercury. This is higher than our typical capillary pressure throughout the body. All right, so you're not going to see this high a hydrostatic pressure in the capillaries in your tissue, in your muscles, certainly not in your lungs. And so part of what keeps that pressure higher is the more narrow efferent arteriole. Like putting your thumb over an open garden hose and having pressure back up behind it. So the more narrow efferent arteriole slows down the flow of fluid, of blood out of the glomeruli, causes pressure to back up. And so we have a higher hydrostatic pressure. Right? Now, we do have proteins present in the blood. So we certainly have our albumin, which is our major osmotic pressure control protein, and those are going to have a pulling effect. Similar to what we talked about with the lymphatic system. Let me make this arrow a little bit bigger because it's almost twice as strong. Okay. So this pulling effect is due to capillary osmotic or colloidal pressure from those proteins. And that's an opposing pressure of approximately 30 millimeters of mercury. And as fluid is pushed into the capsule, some of that is going to flow on into the proximal convoluted tubule, and some will flow, from there to the nephron loop and so on as it moves its way through. But if it's pushed at a rate faster than it flows through, that pressure is going to build up. All right, and so it's like trying to fill a balloon with water, or maybe a plastic sack with water. If there's already water in there, it's going to make it more difficult for more water to go in. And so we have the capsular hydrostatic pressure, which is also an opposing force to our primary hydrostatic pressure. So this would be a capsular hydrostatic pressure.
Your notes say that it's positive 30.
Pardon?
When you put the minus sign in front, it looks like it's a negative value, but you're just.
It's an opposing value.
Like an opposing value.
In other words, this is pushing it into the capsule.
Oh, so it is.
These two are preventing it from going into the capsule. Okay? And so I use it as a negative value because our net glomerular filtration pressure, if we subtract those two values, and our net filtration pressure is 10. Now, obviously, this can be changed.
So wait a minute. So, isn't it 40 here? Am I tripping right now? So 50 minus 30 minus 10. Okay. I'm lost.
Now, if our capillary pressure is high, all right, let's say that you have high blood pressure, and our capillary hydrostatic pressure is high, then we're going to end up with a higher glomerular filtration pressure. What happens if you are not getting an adequate amount of protein in your diet, and we decrease the amount of capillary colloidal pressure? What's going to happen to your filtration pressure?
Increase.
It's going to go up because we don't have as much pushing it back. What happens if we have a blockage to our flow through the filtrate? Maybe we have a kidney stone in the pelvis of the kidney.
Then it just hurts you, really don't feel that much.
And it increases hydrostatic pressure in the collecting ducts, and in the distal tubules, and in the nephron loops, and in the proximal convoluted tubule, and in our capsule eventually. It's like looking at congestive heart failure backwards. So that's going to increase our capsular hydrostatic pressure. And that would also decrease our overall net glomerular filtration pressure. What happens if some people have a genetic problem, they have a mutation, they don't make the right proteins for the slit diaphragm, or they don't have the right proteins in the basement membrane, and therefore they allow albumin to move out of the capillary and into the filtration fluid? Is that going to increase or decrease glomerular filtration pressure? If we take some of these proteins that are supposed to remain in the blood, and we move it into here, then that would increase, we would actually add, notice we don't have a capsular hydrostatic pressure up there, so we would have to add that, and that would increase the pressure, because it would actually start pulling more fluid.
We have a higher filtration.
But would have a higher net glomerular filtration pressure. So all of this filtration pressure is under tight control at the filtration site, at the glomerulus. Right? Yes.
This net hydrostatic pressure is just making the capsular and the capillary.
Right. Right.
Okay.
But at the very end, you would end up with
Okay.
Laura. All right, so you can see here, this is kind of reverse from what I have drawn on the board, or mirror image of what I have drawn on the board. But we have a structure known as the juxtaglomerular apparatus that is involved in what's termed autoregulation of the filtration rate and filtration pressure. We're going to look at this in more detail on Thursday after we've talked about what happens to the sodium ions throughout the nephron, okay, because these changes are going to affect hormones that regulate the distal tubule. And I'm not ready to talk about all that yet. We've talked about aldosterone and ADH when we talked about blood pressure with the heart. I'm going to show you where those factors start to be stimulated to be released, but then we'll finish it up on Thursday. All right, so I'm going to introduce you to some terms and some names and some events, but we won't come to a complete understanding of those actual steps until Thursday. So we already have diagrammed here the afferent arterial and the efferent arterial. And what I need to add is part of our distal tubule. Remember on Friday I said how important it was for the distal tubule to come up between the afferent and efferent arterial? And this is why. So I'll draw a cross-section of it. And typically the distal tubule is formed of cuboidal cells, simple cuboidal cells, with vague, scattered, small, short microvilli. But right at the region where they face the vascular pole, the cells become columnar and slender and tall. This is our description of columnar. And the nuclei become very close together. Okay? So if I may erase this diagram, these diagrams on this side, I'll write a
So
list of
simple
those
columnar,
structures.
simple cuboidal, right?
And so we have this group of cells that form the juxtaglomerular apparatus. Oh, just write it down. Again, this is involved in what is known as autoregulation. So whether you get up or sit down or you're out exercising or you get angry or your blood pressure goes up or down, at the site of filtration, it maintains a relatively constant filtration rate. Okay? So two major two major events function in autoregulation of the glomerular filtration rate or pressure. One is a myogenic reflex. I don't think I have it written out quite like that on your lecture notes, but what we have occurring is, remember what happened to ventricular muscle when it got stretched out? The farther, the Starling's law, Frank Starling's law, the more it got stretched, the harder the recoil and the contraction. So this is a similar stretch receptor reflex. So as we increase, as pressure flow increases and stretches the diameter of the afferent arterioles, they reflexively contract more. All right? So essentially, the afferent arteriole constricts when it's stretched. So an increase of blood flow. So for some reason, there's a transitory increase in blood pressure. We have an increase in blood flow through the afferent arteriole. It responds by decreasing its diameter, and that's going to limit glomerular filtration rate. It's not going to let that rate climb too high. So it's going to limit the diameter of the afferent arteriole. If you stand up suddenly and your pressure drops because of blood accumulating in your lower limbs, all right, blood pressure drops suddenly in the afferent arteriole, those muscles relax and the afferent arteriole dilates, and that keeps the same amount of volume of blood flowing into the capsule. Okay, so that's the myogenic reflex. So it's a responsive or a reflexive